go. So, welcome everyone. Good morning, the team in Pakistan, as well as good morning everyone who is in our time zone. Good afternoon, and we would like to start yet another session of our German club. And the stage is yours. Here you go.
Good afternoon, everyone. So the title of the research article is the angiopoietin receptor Tie2 is atheroprotective in arterial endothelium. And the first question is about Tie2 plus fibroblast. So they show in vitro. Yes, they showed it in vitro in their culture Tie2 plus fibroblast. That Tie2 plus expression in those cells reduce inflammation and hypothesize this may play a protective role against atherosclerosis. Yet in HFD-treated mice, they don't see a reduction, but an increase in number of those fibroblasts. So here is a thing that in that figure, they actually evaluate the role of diet. Like when they given Western diet for 20 weeks, as well as they give PCSK9 overexpression. They are now trying to evaluate the impact of diet. There isn't a role of Tie2 plus receptor here. Here we can see that when they given Western diet for 20 weeks, there is increase in cluster of Tie2 plus fibroblast and other immune cells. But instead, what I depict is this increase in the Tie2 plus fibroblast and the role of Tie2 plus fibroblast in inflammation-related transcripts upregulation is clearly suggesting that a Tie2 plus receptor has some sort of immunomodulatory role, and it is clearly suggesting that Tie2 plus signaling regulates immunomodulatory responses in the Tie2 plus fibroblast. And another thing is, here we can see that these are the genes whose expression is increased. That these all are the genes that are inflammation-related transcripts, like CCL7, interleukin 6, and C3, which are elevated. We can see here that the dot is somewhat bigger in size. This is what happening when they given those mice, like the wild-type mice, Western diet for 20 weeks, as well as here they actually increase the expression of PCSK9. So the question that they are, whoever is asking that, despite this increase, the level of Tie2 in those fibroblasts are lower in normal diet-treated mice. So here, actually, increasing number of fibroblasts has nothing to do with the level of Tie2. Here, actually, we are just trying to evaluate. They are trying to evaluate the impact of diet. And another thing they ask is, do they have experimental evidences? They did another experiment in which they actually reduced the expression of Tie2 in the culture Tie2 plus fibroblasts in order to check whether it has some sort of inflammation-related role, or they are involved in some those inflammation-related pathway or not. So when they actually did some sort of RNA silencing to reduce the expression of Tie2 fibroblasts, when they checked, they actually evaluate that it does have a role in inflammation-related pathway, where they actually see that the expression of those genes is increased. So they actually here they evaluate that Tie2 fibroblast actually does have some immunomodulatory role. Is the answer clear?
Yeah, that was my question, and my question was a little bit different. I saw that they used this silencing method in order to check this Tie2 expression in the fibroblasts and to evaluate the functionality of that. But did they see in vivo that there is a decrease of expression of Tie2 in fibroblasts in those mice which were treated with a Western diet?
No, they actually did not. No, they actually did not check it in vivo. They actually did it in vitro. This experiment was all done in culture fibroblast.
I know, but the point is that, okay, they're looking for something in vitro, but in order to try to validate some hypothesis. So it seems that their hypothesis is that there is the decrease of Tie2 in fibroblasts in vivo might render the system towards like more inflammatory state, which they try to also recapitulate in vitro. But I don't see any evidences from in vivo about that. But the only thing I see is that there is expansion of the number of those Tie2 positive fibroblasts. Kind of, there is more number of those, right? That's what we see from this image. Is it correct?
Yes, it is correct. But here, yes, it's correct. But here they are actually evaluating the impact of Western diet as well as the PCSK9 overexpression. Here they are not checking the role of Tie2 plus Tie2 receptor here. So I don't think so. We can relate it with the like Tie2 plus receptor expression here.
That is my problem. That is exactly what I'm trying to say. That they are making just imagine their logic is okay. Yeah. What they see is that in Western diet they see more Tie2 fibroblasts, and then kind of the hypothesis behind should be could it be that the Tie2 decrease in those fibroblasts might render them towards more inflammation? Then they go and then check in vitro and they say, yes, it is indeed the case. But in vivo, they didn't show this. They don't have any hint that the Tie2 decrease of Tie2 in those Western diet mice or like in any pathologic conditions or, for example, in humans. So that is a little bit my problem. So they did kind of, after doing this in vitro experiment, they didn't come back to in vivo in order to prove, yes, indeed, in those Western diet treated mice, we see decrease of Tie2 expression in those Tie2 positive fibroblasts. That was my problem.
Yeah. Actually, here's the thing that despite giving like a Western diet for 20 weeks as well as giving like PCSK9 overexpression, we could not expect that Tie2 receptor could outperform its function in like reducing the inflammation, because diet is another factor. Like we could not say that Tie2, if we are constantly giving Western diet and we are increasing the expression of PCSK9, and we could expect that Tie2 receptor could outperform its function in reducing inflammation, because diet is another factor.
Sure, but
yeah.
Okay, but then what is the point? I mean, is it only genetics? Is it only to see that there are some kind of mutations which are associated with the Tie2, and then we are kind of trying to find out that this might have some pathological role.
Actually,
because there are some studies where they show that Tie2 is being depleted, or at least the expression is decreased in case of like the shear stress, right? So it was in the case of some pathologies of the blood flow or whatever. I think this was mentioned in the introduction. So I think what they were trying to say with this paper is that in general Tie2 is kind of anti-inflammatory. It's kind of protective, and that the decrease of Tie2 might be causing these pro-inflammatory effects, and that is why, I mean, they could have checked the maybe it's not true, but they could have checked at least that the levels of the expression of Tie2 in those fibroblasts. I think that would be logical instead of just showing.
Yes, you are absolutely right. Actually, what I think is, like you are referring the literature, those all were the contrasting results. Which actually like inspired them to explore the function of Tie2 in atherosclerosis development. this is what I actually depict from their like this was their hypothesis actually to explore the function of Tie2 in atherosclerosis development.
Exactly. Yeah. Yeah.
Yeah. Okay. So the next question is, okay. So the next question is, did they compare the aortic endothelial cells of HFT treated versus normal diet-fed mice? Yes, madam, we can hear you.
I was trying, but I could not, my voice was not communicated to you. Can you please go back to the last slide, Javeria?
Yes,
I want to add something, please.
Yes, please.
Actually, in this paper, they are actually trying to elaborate the importance of Tie2 expression as well as diet in causing the atherosclerosis. Like if the Tie2 expression is good and it is atheroprotective, and similarly, the normal diet is good as atheroprotective, you can say. If you can see in this picture, both of the cells actually they were feeding the mice with western diet and normal diet. There were two groups. So this is the in vivo experiment. It is not totally in vitro. They fed the mice for 20 weeks with normal diet and then with western diet, and they checked the clusters of the cells in those both groups, and they saw that although the Tie2 expression was normal in both the groups, but in western diet group you can see the increased expression of these increased clusters of these cells which are mentioned here. So actually they just want to mention that even only in response to western diet fed mice, there is increased number of these Tie2 fibroblast as well as mfab fibroblast. Just they want to say that only western diet also has a role in increasing the number of Tie2 fibroblasts, which have a very important role in modulatory role in either atherosclerosis protective, or any other modulation. So in this picture they just want to, it is the in vivo experiment, it is not totally in vitro. They fed the mice in vivo, and then they after feeding them for 20 weeks and for a particular time, then they check the cluster of the cells. So it is not totally in vitro, it is in vivo experiment.
I totally agree, and I know that. Huma, thank you for your comment. The point is that I want to just separate two points. One is the number of Tie2 positive fibroblasts, and the other point is another characteristic is the expression level within those fibroblasts. That is what I wanted to see, because what we see here is expansion of the Tie2 positive fibroblasts in vivo. Yes, we can see that.
Yeah.
And kind of this kind of opposes. It's like we can speculate, but maybe it could be a compensatory mechanism. Yeah, so you have this Western diet, and these Tie2 fibroblasts accumulate in order to exert their anti-inflammatory effects. Yeah, so this is what we can speculate, and this is in vivo. So that's nice. My question is, can it be that those accumulating Tie2 positive fibroblasts actually have lower Tie2 expression than the ones which are in the normal diet, and by that it kind of aggravates and that this basically can contribute to the increased inflammation and atherosclerosis. That was actually my question.
Yeah, I personally feel that they could have planned another experiment, but they should have given the Western diet and normal diet to the mice, both of the mice groups, and they should have checked the expression of Tie2 in both of these groups. But they did not did such experiment. I mean, they just this experiment is missing, and I would say that this is the limitation, a bit technical limitation of this paper, that they should have checked the expression of Tie2 in both of these groups, but that data is not available in this group. I totally agree with you in this regard, but actually I would just say that actually in this paper they are showing both of the factors, that Tie2 expression is good, and as well as normal diet is good if you want to avoid the atherosclerotic process. So this is total theme of this paper.
which is also not expected, right? Not unexpected, yeah. So this is something which is they just nicely show the mechanism of one of the aspects of that, right?
Yeah, thank you.
Should I move forward?
Hmm.
Okay. So the next question is, did they compare the arterial endothelial cells of HFD treated versus normal light-fed mice for Tie2 expression? So the answer is no. In this study, they did not compare arterial endothelial cells of HFD treated mice versus normal light-fed mice for Tie2 expression. And what I think is there wasn't a need for that, because main purpose of Western diet and the PCSK9 expression was to develop sclerotic mice model. If the mice is normal, then why would they compare the Tie2 expression Tie2 expression between the normal diet fed mice versus HFD treated mice? And another question is, had it been shown also for humans with developing atherosclerosis or obesity? So actually this study says Tie2 function in atherosclerosis was unknown. So they are actually trying to say that they are the first one who are providing the evidence that variation of arterial Tie2 expression is associated with the development of coronary atherosclerosis in human cell lines, HAEC, and they actually identified that the risk variant is located within a tumor necrosis factor enhancer element, which is linked with through the chromatin interaction with the Tie2 promoter. And what they did, they actually CRISPR-mediated deletion of the enhancer region, which led to the significant decrease in the Tie2 messenger RNA expression, which is providing additional evidence of its functional effect. Okay, so the next question is, tamoxifen treatment was intraperitoneal or intravenous? So the tamoxifen treatment was neither IP nor IV. They gave tamoxifen treatment via intragastric doses for the induction of pre-mediated recombination in mice. And the next question is, can you please explain?
Excuse me. Intragastric means they have per oral tamoxifen. They have given it orally. I really doubt that. I think why would they do that? I think bioavailability of tamoxifen is by IP is close to 100%. It's comparable to the bioavailability of the intravenous one. So I would really be surprised that they do it intragastric, or maybe they mean that by IP they mean intragastric. I would be really surprised if it is really true.
Atherogenic diet, and then they gave chlortonate to decrease the macrophage.
Sorry.
Yes, please.
I think you missed my comment here, right? So because I think you dropped, or because I was asking about the tamoxifen, the previous slide. So I was just saying that I was really surprised. So if you mean that it has been delivered intragastrically, means what? Means per orally, like a gauge? Are you sure?
It means, yes, they actually mentioned in this research article, they gave it via intragastric route.
Intragastric?
Yes.
Why would they do that? I mean, the bioavailability of tamoxifen, like orally, is lower than IP or IV. Why would they do this? I don't quite understand. I'm really surprised. Like we usually deliver tamoxifen IP. That is a very common way. That bioavailability is very high. So where is it? It's in the methods, right? Should be. I mean, it's a minor detail. Like not probably not so important. But I'm just I hear it for the first time that someone delivers it orally. Let me see. Yes.
Can I ask about tamoxifen? I don't know much about this drug. I mean, I know that tamoxifen is used to treat memory cancer. And so why did they choose this drug here? Can you please explain a bit?
Yes, they actually at first they crossed two mice. One was a mice in which the exon 1 was flanked by the LOXP site, and another mice was BMX-CRE-ERT mice in which there was one BMX promoter and then CRE-ERT in which, and then estrogen binding domain of there was an estrogen binding domain. So when CRE recombinase is expressed, it is actually in inactive form. There is a need for some drug from the outside tamoxifen in order to turn it into active state. When tamoxifen, yes, then they induce the CRE-mediated recombination.
So in short, the Hitchcock protein 90 is binding to this domain, to the estrogen receptor. And then it will dissociate only upon binding of its ligand, and its ligand is tamoxifen, which is a blocker of the estrogen receptor, but it is this CRE-ERT2 is a modified version of estrogen receptor. So it recognizes specifically this tamoxifen. And then upon dissociation from the HSP90, this whole protein, because it's fused, the CRE-ERT2 is fused to the CRE is fused with this estrogen receptor domain, goes to the nucleus, and then since it must exert its effects in the nucleus, so that's when. So you basically can control when it happens. So you can dictate when you want the patient to.
Yes, they actually temporarily control the silencing of this gene.
In fact, I just wanted to comment. I think these guys probably just mistyped. They wanted to say IP, I guess. I don't think they have done it intragastrically.
Maybe
I would be.
They did it intragastric. Dr. Elia, they did it intragastric, but I personally feel that intragastric is different from oral route. I mean, they just they need a needle and directly inject in the gastric, stomach and all the. It's not oral route.
I know, but the bioavailability will be the same. I'm just saying that I know it's not the same, but bioavailability of oral route or intragastric route would be the same. My point is that injecting IP is much easier. so and you get 100% bioavailability by IP. I don't know why would you do intragastric. I suspect that these guys they just mistyped. They wanted to say IP, but they just wrote it in the intragastric because it's also somewhere there near the belly. That's what I suspect. But I never seen anyone doing the intragastric injections of tamoxifen. That sounds really strange for me.
Yeah.
Maybe they might have made a mistake while writing.
No, I don't think so, Joy.
But maybe, I didn't Google it. I didn't Google. Maybe some labs do this, but I would be really surprised, as I said. So IP. Anyway, if you are going to do this, do it IP. It's very easy.
Okay. Thank you for the information. Okay. So the next question, can you please explain the setup of fluoronate fluorospride anti-CD11B experiment in more detail? So actually what they did, they gave tamoxifen when the mice was born, and then at the 3rd week they gave PCSK9 overexpression and treatment and the Western diet at the 3rd week. And at the 7th week, they give fluoronate to deplete the macrophages for the normalization of those immune cells. And then after after 3 days, they give fluorospride in order to label those circulating monocytes, because they wanted to explore the adhesion of these circulating monocytes in a real time via intravital microscopy. And then after 11 weeks, after 12, 13, they gave antibody CD11B to bind with the myeloid cells, and then they explore its adhesion at the bifurcation area. Here we can see that when they delete it, there is more binding of those myeloid cells at the bifurcation area, and here we can see that those circulating monocytes are adhered to the aortic walls. So the next question is
wait a minute, please. Thank you very much. Yeah. So again, Jawaria, thank you. So just to be clear, because I'm not familiar with this technique. So they use first this clodronate in order to deplete all the monocytes, right? Is it correct?
No, they use chlorinated to deplete macrophages, like resident macrophages.
Macrophages. Okay.
Yeah.
And then they like this fluorescent bright would bind what exactly?
To circulating monocytes.
Only to monocytes. So this fluorescent bright would only enter the monocytes, right?
Yeah.
So
maybe they bind to the marker present on the monocyte.
They will what? Say it again.
I said maybe fluorescent bright microspore. No, you are absolutely right. I thought that it might have bind on the marker on the present on the monocyte, but now I remember that it actually engulfed into those circulating monocytes, and then they visualize in a real time the adhesion of those circulating monocytes to the bifurcation area of carotid artery.
Okay, so clodronate was done kind of IP or IV kind of it was delivered systemically, and also those fluorescent bright, was it done systemically?
IV.
Is it IP or IV? IV probably,
IV.
right?
IV intravenously.
Both clodronate and fluorescent bright, right?
Yes.
Okay, then we'll label all the circulating monocytes, you say, right?
Yes.
With fluorescent bright, it enters inside the monocytes, and they are getting labeled, so they are somehow yellow. And then after how many weeks? Like 4 weeks, it seems. They do intravenous IVM anti-CD11B. What is IVM? Intravenous what? I don't know.
This is the IVM anti-CD11B, which actually binds to the markers present on the myeloid cells, which is CD11B. They further evaluate, or we can say validate that either in order to visualize in real time, either they are binding to the carotid artery or not, like myeloid cells. In this case, they actually induce this antibody to bind with the marker CD11B, which is present on the myeloid cells. This antibody actually binds with the CD11B markers, and they actually visualize it in real time.
Okay, wait a minute. What yellow are monocytes now, right? Monocytes are yellow because they have engulfed those fluorescent bright, and they were circulating there for 1 month, correct? Because on 7.5 weeks, we labeled all of them with this fluorescent bright, right? So they are kind of, we have yellow monocytes in our bloodstream, and all the macrophages were kind of depleted by clodronate beforehand. So we don't have like the macrophages in the tissue. So we are checking the new novel, like the monocytes which were recruited to the sites of some injury, right, in the course of this 1 month. And then in the end, we use. Explain again, please, what is this anti-CD11B antibody, and how do we visualize? Is it coupled with some fluorescence as well, or like what is the how's it being
I think it is coupled with some fluorescence. Otherwise, they could not have like visualize it. Like it must be coupled with fluorescence.
Okay, I don't think so. Actually, I think this fluorescent bright is actually green, and what we see on the left. Which was probably not shown in the main figure, but probably somewhere in the supplement, is those circulating monocytes.
Yes, and the green one are the circulating monocytes.
Yes, and we see that they are starting to accumulate on this bifurcation. So, and they suggest that that it seems that there is something which attracts them, so there is increased inflammation there. And this CD11B was done just ex vivo. Basically, they already stopped the experiment. They treated with this anti-CD11B, and they just visualized that there is accumulation of those monocytes in this bifurcation. So kind of independently showing that like fluorescent bright was not enough. They want to make sure, this is really CD11B, which is accumulating. That's just yet another way to identify. This is at least my interpretation. Correct me if I'm wrong.
No, you are absolutely right. They actually, in this case, they actually exposed the carotid artery. Then they like administered those IV, NTB. Like you are absolutely right.
Okay, then that is great. So then we resolved it. Okay. Thank you.
Is a SNP a nonsense or missense mutation? So the answer is this SNP is an intron variant, and intron variants themselves do not cause missense or nonsense mutation because intron are not a part of coding sequence. And the another question is C allele associated with increased Tie2 expression conferred reduced risk of coronary artery disease, which is very interesting. Can you please provide more details? Actually, in this study, what they did, they actually took the advantage of meta-analysis of multiple JIVA studies for approximately 1.5 million individuals to improve the candidate gene discovery and to examine the association of this variant and of the Tie2 gene with the cardiovascular disease. And here they actually find that this top associated variant is significantly associated with the expression of Tie2, which is suggesting a functional effect of this SNP. Okay, so the next question is CRISPR-mediated deletion of the enhancer region of the Tie2 was done in human cells. Yes, CRISPR-mediated deletion of the enhancer region of Tie2 was done in human cell line, TLO-HAECs. And could you please provide more detail about this experiment? So actually, they determined that this SNP is localized to the cis-regulatory enhancer element, and that is a tumor necrosis alpha responsive enhancer region, which is loops to the Tie2 promoter. And they did CRISPR-mediated deletion to study the effect of this variant on Tie2 expression in human cell line, aortic endothelial cell line. For what they actually use 2 pairs of guide RNAs for to make the deletion at 945 base pair and 1120 base pair region centered around this SNP variant. They actually supplied those. They actually gave those CRISPR component by the electroporation technique, and then they did the quantification of DNA band intensities on the gel image. Here we can see that in the left bar, there is approximately 80% deletion of those intron variants, and this deletion is translated to like 30 to 35 to 44% reduction in the expression of Tie2 or TEC gene.
Okay.
Okay.
Thank you. Jawaria, can you get back to the previous slide, please? I just wanted to make sure that okay, so for this intron variant, so is it known whether it is kind of impacting the intron the exon skipping, or like some alternative splicing, whether it is like somewhere in the intron acceptor or donor side, like, or is it known whether this mutation specifically is related to this?
Actually, it does have some sort of regulatory role, because this SNP actually lies in a region near the tumor necrosis enhancer factor, which through the chromatin interaction and lies near the Tie2 promoter.
I can a little bit add, Emilia, that in gene regulation, not only the enhancer, promoter, or exonic regions are important, and as you mentioned that exon skipping can influence the formation of that respective protein, but also the intronic regions are other non-coding regions where some transcription-related machinery binds. So in this case, the allele that is top candidate, C allele, that region, that particular SNP is having space for binding of TNF enhancer region, which loops with the enhancer region or the promoter region of this Tie2 gene. So in that way, the gene expression of this Tie2 gene is influenced by this variant. And if that allele is present, then either that is expressed at high level or low level, that influences the availability of Tie2. So in gene expression, I mean, the non-coding variants could contribute, as this is the case in that one. And this is not mutation, that is polymorphism, and that has been studied in like various populations, and then they have conducted the meta-analysis comprising about 1.5 million individuals. So they have great confidence that this is contributing towards the expression changes of this Tie2 gene. And based on this knowledge, then they have devised that CRISPR-mediated deletion of that region to study the downstream functional impact. So that is what I understand about this. Thank you.
Yeah.
Yeah, thank you very much, Fazli. I totally agree. So that looks really, really interesting. That's why I think it is pity that they didn't include this to the main story, because that part is especially interesting for me, because like it looks like they have this link, and they really also nicely show that this deletion of this enhancer really directly impacts the expression of this gene. So that this kind of looping and those kind of enhancer regions within the intron, or the regions which are looping with the enhancer, that is, I think it's very, very interesting. So it's I think it's a strong part of the story.
Yes, I agree. Endorse. So I think Jawaria is done. Yes, please.
Yes, please.
Jawaria, would you like to make kind of a summary or a kind of conclusion? Like, what is your own attitude, maybe on the design, the hypothesis, or like the way how they try to use the methods and so on and so on? Like the overall, like how do you assess this story?
actually, I personally find this study so magnetic. Like they are the first one who actually explored the function of Tie2 in atherosclerosis development. Because in the previous studies there was a role of Tie1 receptor only explored. There was an interaction of Tie1 with the Tie2 was explored in the previous studies. So I personally find this study really amazing. Like. This increase further insights into this study.
Any other comments, any other suggestions or questions or opinions?
No.
Zishan?
So I
Hello,
think
Zishan.
I want to
further,
add
Yeah,
something.
sure.
I think further studies are needed to see the Tie2 expression in real human samples, whether in which bypass experiments, coronary bypass experiments are done to see how that is impacting the atherosclerosis development, the real evidence from that, and then associating that with this polymorphism, the expression levels. So that will be interesting, I think, in future.
I would want to add something. I mean, when I was like reading this paper, I want to add a bottom line of this paper, that genes is not in your control. Your genetic content is not in your control, but your habits are in your control. So after reading this paper, I came to the conclusion that this SNP, whether it is C allele or other allele, it is not in your control. It is impacting the expression of Tie2, and if the expression of Tie2 is good in you, you are at a protective end. But you can change your diet. In many results they are actually telling that not only the Tie2 expression is good, but also the normal diet or good diet is important. So we cannot change our genetic makeup, but we can change our habits. So in this atherosclerotic model, actually they are using the Western diet as well as the Tie2 knockdown. So I would not sorry Western diet, and so I just want to add that genetics is not in your control, but you can control your diet to beat the wrong genetics in you can say.
And that to that point, I mean, the whole point of investigating Jiva studies is to identify candidate gene or genetic variants, so so that these can be translated into personalized medicine. Because if a person is having snapshot of what genetic variants are predisposing to such kind of diseases like coronary heart disease, atherosclerosis, or stroke, then only they could be very careful which kind of diet or lifestyle changes they have to modify based on their genetic makeup.
I agree.
Thank you for your really great insights, Fazli and Huma, and also Jawaria, of course. Like really great presentation. The first video which I watched was really beautiful, really nicely structured. Also, like you nailed from the very beginning, like the take home message of the whole story. So it was so easy to follow. Overall, really nice. And also Huma's comments, they are really awesome. They are just, she goes like more like philosophically. She's trying to make sense of the big picture. I really enjoy it. Zishan, do you have any suggestions, or maybe Ayman or maybe Nastya? Do you have any comments or any?
I just wanted to say thank you. Yeah, it was very nice. I like the presentation and like the paper. I think it's very awesome. Yeah, thank you.
Yeah, I agree. I actually I didn't tell my opinion about this paper. I think it's a very strong one. I really think they have done a good job, and it is also logically and very well structured. Really nice, really good job. And also Jawaria, also you're really great work. It was really a pleasure. And overall, it was actually a pleasure to work with you and to partner with you this semester. It was a really nice ride. I think we learned a lot. We really learned a lot from you, and from interacting with you was really great. Pleasure. So it was so nice.
We are indeed, Elia, thank you very much. We are indeed very grateful for providing us this opportunity through this quiet program. My students, they have, you made them really think deep, and they are developed into deep thinkers, how to analyze studies and how to present their studies. And your positive feedback and criticism that basically illuminated their minds towards scientific discovery in future. So we are really grateful to you and your lab members, all of them, for providing this platform and interacting with us and teaching us so many good things. Really, we all are thankful. Thank you.
Thank you. The pleasure is all ours. Thank you. Zishan, any comments from you? Zishan, are you now with us, or anybody?
Suggestions. Thank you for the structured presentation.
Okay, so with that, I think we can wrap up, right? And we say Happy New Year to everyone. To have a really fruitful year to everyone, and I hope at some point we will be able to also communicate and also share some scientific knowledge later as well. So it was really great having you. Thank you very much.
Thank you.
Yeah, Happy New Year to you all as well, and have good New Year, and all the best for this New Year, and hopefully we will catch up again in near future after several weeks break now. Thank you.
Thank you
very much.
for these brainstorming sessions.
Bye.
Thank you.
Bye.